right, we're going to give a couple minutes for people to join on. Okay, let's get started. I'm sure more people will jump on in as we go, but first things first, I'm Maria Vogel. I'll be one of our hosts today. I work at the Atlanta Botanical Gardens in our conservation and research department, and Amanda's joining me as well. She's one of our lab managers. And yeah, this is Science Cafes are a monthly speaker event that our department puts on to um, further educate people on um, different subjects related to plant conservation, genetics, things that we do within our department, and to spread the good word of all the amazing things that are happening with a lot of our partners, previous advisors, and connections. So this is one of them here with Dr. Maggie Wagner. And I'm going to actually have Amanda introduce Dr. Wagner today.
Awesome. Thanks, Maria. So yeah, like she said, I also work at the Atlanta Botanic Garden in the Conservation and Research Department. I'm the Conservation Genetics Lab Manager, and I'm very excited to introduce Dr. Wagner today. So Dr. Maggie Wagner earned her BS in plant biology at the University of Michigan, followed by a PhD in genetics and genomics at Duke University. Her dissertation research focused on the ecological causes and evolutionary consequences of phenotypic plasticity in a wild perennial mustard species. As a graduate student, she developed strong interest in plant-associated microbiomes, both as part of the plant's environment and as an extended phenotype of the plant host. She was awarded an NSF Plant Genome Postdoctoral Fellowship to investigate how modern breeding affects the composition and function of the corn microbiome while based at North Carolina State University. In 2019, she established her lab in the Department of Ecology and Evolutionary Biology and the Kansas Biological Survey at the University of Kansas, where she continues to work on the complex interplay between plant genotype, phenotype, and microbiome. And I just want to say I had the pleasure of working with Maggie at KU, and she's just so amazing. Without her mentorship, I don't think I would have gotten to where I am today. So I'm just really excited for her to be here and to share her amazing work with y'all. And I'll hand it over to you, Maggie, whenever you're ready.
All right. The host disabled participant screen sharing. Maybe Maria can help with that. In the meantime, thank you, Amanda. Yeah. Really nice introduction. Amanda was such a champ working in my lab in the summer of 2020 when nobody else was allowed to go in the building because of the pandemic. And so she was such a trooper doing like hundreds of DNA extractions by herself in the lab all day. So, and yeah, we're starting to publish that work now based on Amanda's hard work. So very exciting. Atlanta Botanic Gardens got a good one. So I'm going to go ahead and share my slides. Are you seeing the correct thing?
Yep, I think so.
Great. Okay, well, I want to thank everybody who is in attendance here at the Science Cafe. I saw someone shouting out hello to all the fellow plant enthusiasts, so hello back. I'm excited to talk a little bit about some of the research that is currently going on in my lab. And specifically, the topic that I'm going to focus on is sort of the evolutionary relationship between plants and their microbiomes. And I'll explain a little more about that and why we think they're important for each other in just a moment. And just as a bit of a teaser, this image that's on your screen right now, this is a cross section of a maize root. And I think it's really beautiful personally, but you can see all the different layers of cells and the really intricate structures. This part in the center here, this kind of circular part, is where all of the water transport up and the sugar transport down happens in the plant. And so you'll see this figure again a little later in the talk. So I just want to set the stage a little bit. This mainly, I work on sort of agricultural problems as well in the field of plant microbe interactions, but what I'm talking about today is evolution. And so we know that, like a lot of different organisms, plants have to be able to adapt to stressful environmental conditions. And for plants, a really big one is the availability of water. And you can see this really readily when you think about a very humid climate like you all have around Atlanta, lots of green plants during the summer, very lush environment, as opposed to the more desert-like environments that you see in drier locations. So you notice both a difference in the quantity of plants and also the types of plants. And just like the dry conditions will select for adaptations in plants, they will also select for adaptations in the microbes that live in the soil. And I'll say a little more about that in just a moment. So I want to start by introducing, just at a broad level, the concept of a microbiome. So many of you may have heard something about the human microbiome, as in the past decade or two, this has really become a hot topic in the medical field, because it's becoming apparent, the more we study this, that the really complex communities of bacteria and fungi and viruses that live on and inside our bodies can have a really strong impact on many different aspects of our health. And so just like us humans, plants also have microbiomes occupying every part of the plant. So this image is showing the, looks like mostly bacteria with a couple fungi that are resulting on a Petri plate after pressing a tomato leaf onto the plate. And so you can kind of see how the leaf's outline is being, represented by the bacteria that grew just out of the surface of the leaf. And so the organisms shown there are just a very small subset of these really complex communities. It turns out that below ground, the plant microbiome is even more complex than the above ground microbiome. So you have in the roots of the plant and in this special area called the rhizosphere, you get the most diverse microbiome of all. So the rhizosphere is this thin layer of soil that is sort of coating the outside of the root. And we distinguish between the rhizosphere from kind of what we would call the bulk soil, because the plant has a big influence on the rhizosphere environment in a way that really matters to these microbes. So for example, all the sugars that the plant makes through photosynthesis up in its leaves, a lot of them get transported down. Some estimates suggest up to 50% of the sugars that are generated by the plant through photosynthesis end up being put back into the soil through the plant roots. And those sugars are a major source of food for the microbes living in the soil. And so what you end up with is this rhizosphere layer of soil surrounding the root, which is a much richer environment for these microbes because there's so much more sugars there for them to eat. As a result, you get very strong microbial population growth in those areas. And you also see kind of a subset of the root micro or the soil microbiome, which is able to specialize in the rhizosphere. And so the numbers of microbes that are on or inside a given plant are really staggering. So we're talking billions and billions of microbes here within an individual plant. And critically, why we're so interested in this, even as plant biologists, which is what I would consider myself as, it turns out that these microbes living on and in the plant can have really strong effects on the survival, the reproductive success, and the overall health of the host plant. One of the reasons for this is because when you think about thousands of different bacterial or fungal species living on a plant, think about all the different genes that each of those individuals has. So like a typical bacterium might have somewhere around 4,000 to 10,000 genes, something like that. When you add up all those genes together, you end up with a lot more genes than the plant itself has. And what this means is that this really increases kind of the functional potential of the plant to the extent that these microbial genes can perform functions that are important for the plant. And so at a kind of a higher level, these are some of the benefits of a well-functioning microbiome for the host. So phytostimulation, just another way to say kind of like growth increase, acquiring nutrients from the soil. So a lot of those bacterial genes can sort of, they produce enzymes that will help to process nutrients in the soil to make them more easily absorbable by plants. Some of them can directly suppress pathogenic microbes. So in addition to the sort of beneficial component of the microbiome, there are also natural enemies like disease-causing organisms. And same with natural enemies that are insects or nematodes. And finally, the focus of my talk here is going to be abiotic stress tolerance. So there's a variety of mechanisms that these microbes can provide to help the plant weather stressful situations. And so one of those is simply, for example, being able in the root to activate the plant's immune system in a way that increases its resistance to pathogens. And also the nutrient uptake that the microbes help with can lead to sort of like an overall increase in the plant vigor, which makes it better able to withstand a lot of stress. But there's also some more specific mechanisms. For example, a lot of soil dwelling microbes can actually produce hormones that the plants also produce. So just like us humans, we have hormones that are sort of regulating basically all of our bodily functions and growth and so forth. Plants have a lot too. And so these microbes that are able to either produce directly these plant hormones or are able to, for example, degrade the precursors of those plant hormones, things like that. In that way, they can alter the hormone status of the plant and have potentially really big effects on the plant health. And that includes being able to alter the levels of stress hormones, which is how that abiotic stress tolerance happens. And as a result of all these beneficial functions, there's been a lot of interest in the agricultural sector about how can we sort of harness the power of these beneficial plant microbes in order to have better producing crops and crops that are more resistant to sort of, for example, extreme weather events or degraded soils, things like that. This is part of the overall effort to get kind of more sustainable agricultural practices. Now, the problem is that these plant microbiome systems are really, 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 really complicated. So for starters, we're talking thousands of different species of bacteria and fungi, et cetera, living on the host. You've got all kinds of crazy ecological interactions going on, not just between the plant and the microbes, but between the microbes themselves that are within this community. And so you have, those organisms are fighting with each other for resources, or they might be producing kind of byproducts that other microbes eat, and so they can also have positive effects on each other and so forth. So there's pretty much, limitless moving parts, which make it very difficult to figure out how exactly can we modify these plant microbiomes in a way that is beneficial to us. And the part of this problem that my research focuses on is the role of the host genotype in the assembly and the function of a microbiome. And I'll explain a bit more about how this works. So the plant microbiome starts in largely from the pool of microbes that are living in the soil. And then the genetic variation in the plant. So for example, think of two different cultivars of corn, or maybe like a drought adapted plant versus a non-adapted plant. Those genetic changes in the host can actually trickle down to affect which microbes end up colonizing the roots. And so at this point, I like to show these images because they really, I think, help us understand how it is possible for plants' genes to affect their microbiomes, because the way genetics works, it's not possible for a plant gene to produce a microbial cell, right? Plant genes make plant proteins and plant cells. And so how does this work? Well, take a look at these photos. So these are scanning electron microscopy images of the surface of a leaf with a single species of bacteria living on them. So the left and center panel have a beneficial species of bacteria called Burkholderia carabensis. And on the left, you can see that these bacteria are occupying the crevice in between two pavement cells on the leaf surface. And then in the center panel, those bacteria are colonizing the area surrounding a stomate. So the stomata are these pores on the surface of a leaf, and that's where gas exchange happens to enable photosynthesis. And these stomata are also a common point of entry for bacteria into the interior of the leaf. And on the right, we're again looking at the crevice between two pavement cells, but this is a different bacterial species. This is a Pseudomonas species. You can see they're nice rod shaped. And you can see all these like kind of web-like structures between them. Those are the bacteria forming a biofilm in that sort of crack between two plant cells. And so the point of me showing all this is because now think about different types of plants and think about how they can vary in traits that might really affect the habitat that these microbes have to colonize. So for example, like the topography of the leaf surface, or the behavior of the stomata. So, the rate at which they open and close, the stickiness of the leaf surface, things like that. And the same is true in roots. So here is a scanning electron microscope image of a single root, and these tiny appendages coming out are the root hairs. And that's where the majority of water absorption happens. And also remember, there's a lot of carbon-rich compounds being dumped out through the roots and into the soil. So it's easy to imagine how kind of this physical structure and the chemicals being provided through the roots will be really important to the habitat quality for these microbes. And so what we end up seeing is kind of this funneling process where we start with, in bulk soil, we have what is the most microbially diverse environment known on Earth, actually. And then when you start to look in the rhizosphere soil, which is this area of soil immediately surrounding the roots, and remember, it's a richer environment, you end up with a subset of those soil microbes that become abundant in the rhizosphere. And then when we zoom in even further to the inside of the root, that's where things like the plant's immune system and other plant traits become important as well. And so you see an even tinier subset of the original soil community is able to colonize what we call the endosphere, or the inside of the root. And the upshot of all this is that we can actually think of plant microbiomes as sort of an extended phenotype of the plant. So this is what it means for genetic variation in the plants to result in microbiome variation. So if you imagine a plant forming its root microbiome out of this more diverse bulk community, that process is influenced by the phenotype of the plant, so which is represented by this funnel here. And this phenotype represented by the funnel is going to be a product, partially, perhaps largely a product of the plant's DNA, okay? And so as a result, you can have two plants growing right next to each other in the same microbial environment, but if they have different genes that affect traits that the microbes care about, those two plants will end up with different microbiomes, okay? And where it gets really interesting from an evolutionary perspective is when we start to think about how the plant microbiome can, as we talked about earlier, can have really strong impacts on sort of the fitness and health of the host plant. And when we think about sort of natural selection and evolution, that reproductive fitness is all important, and survival. So when organisms are evolving by natural selection, the genetic variance, so for example, this blue variant of the gene versus this gray variant of the gene, the relative frequencies of those two variants is going to change depending on the reproductive fitness of the organisms containing those genes. And so we could have a situation where the main function of these genes is to influence the microbiome through some sort of intermediary trait. So let's say, for example, the plant's immune system. And then if that ends up creating a microbiome that is beneficial, it will actually increase the plant's fitness and feed back to cause evolution in this gene, which is pretty cool. And so there's a ton of different plant functional traits that can be influenced by these microbes. So things like photosynthetic rate, the sort of the structure of the root system. So for example, the angles that the different roots will branch off of each other at, things, and like I mentioned before, the hormone status. All of these are really important traits that affect the way a plant interacts with its environment. And so these are the types of mechanisms that can cause this evolutionary feedback between the plants and their microbiomes. And so this is an example of a concept called the extended phenotype, which has been around since the 1980s. The idea being that genes, through their direct effects on a phenotype, can have sort of cascading effects into the environment. And so the selection of a microbiome would be one example of that. And when this extended phenotype influences the fitness, this can actually cause the evolution of things outside of kind of the focal organism. But there are other ways too that microbes can affect the evolution of plants. And one of those is what we've called phenotypic plasticity. And what this is just any part of the environment can influence the expression of the plant's genes into the plant's phenotype. So you can think about, let's imagine you have two genetically identical plants. One of them is growing in a super rich soil and one is in a very impoverished soil. Even though the plants are genetically identical, their phenotypes are going to end up very different because the environmental context affects that relationship between genotype and phenotype. And microbes can be part of that environment that causes changes in the phenotype. And microbes can also cause natural selection itself on the phenotype. pathogens are a really simple example of this, but you can also get more complex relationships where the microbes are changing sort of the adaptive value of a certain trait. And so in addition to this sort of circular feedback in in evolution between plants and their microbiomes, it's important to remember that there's also evolution happening just in the bulk soil community, where, you know, we have these complex communities of microbes, and all of them have to survive, and natural selection is happening there as well, even without a plant present. And within, even inside of a plant, another fun thing to think about is that a lot of bacteria in particular have very short generation times. And what that means is that you can get many, many generations very quickly, and each generation is an opportunity for natural selection to change the frequencies of different alleles or genetic variants. And so particularly when you have a long-lived plant, you could potentially get a very large number of bacterial generations within the lifetime of a single plant. And so one thing I'm interested in is how microbial evolution happens within the context of living on a plant or inside a plant, or living far away from a plant. So from a microbe's point of view, the soil itself is a perfectly complex habitat, right? And so you can imagine these microbial lineages, part of their time is spent just in the soil without any plant nearby. These are SEM images of different types of soils. And you can see that there's plenty of different niches or habitat types available to microbes just in the soil. And so I'm curious about, you know, how does that evolutionary process work for these microbes, either within a host and or, you know, completely without a host, just in the soil. And so how does that affect microbial evolution? And then how does that feed back to affect the plant? I'll skip through some of this, other than simply to sum up by saying that this is a hot topic in evolutionary biology right now. There's a lot of debate over the extent to which natural selection can only act on each individual species separately, or whether it's possible for evolution to act on kind of these whole conglomerates of organisms, which some people will call a holobiont. But I'm not going to get into the nitty-gritty details of those competing models right now. Okay, so thinking about these whole communities that consist of a plant and their microbes, again, they're very, very complex. And so how can we tell how these systems are evolving? Well, the project I'm going to talk about today really looks at the emergent properties of these these communities. So things like, you know, when you think about the entire community of organisms represented in this root system here and in the soil that's clinging to it. So this is a rhizosphere surrounding the roots. When you think about that, you can think of like, okay, that's a community, and we can actually measure how diverse that community is. We can also measure, you know, what are the, you know, most important types of organisms that are living in there. We can even measure the total suite of microbial genes that are present in this root system. But what I'm going to really focus on is we can also measure this community's effects on a plant. And so I'll walk you through an example of how we do that experimentally. Okay, so the overall project that this work is a part of is related to water stress. Okay, so we're interested in plants' adaptation to water and microbes' adaptation to water, as well as how those processes interact with each other. So I'll just give a shout out to our team who's been working on this, and especially Nicole Ganan. She was a postdoc in my lab until quite recently, when she went to take a permanent job at Penn State. But anyway, most of the data that I'm going to show was generated by her. Got the beach ball. We got to wait for PowerPoint to catch up with me. And so I'll just start to set the stage a little bit for the study system where this work was taking place. So we're located in eastern Kansas, and Kansas, being right at the center of the country, here we go, is located, it spans this really steep precipitation gradient. And so we get here in East Kansas, where, let's see, this is my county here. In East Kansas, it's pretty lush, and we get a little bit of this eastern deciduous forest, but it's starting to transition into the tall grass prairie dominated area. And as you get farther west, it really gets pretty desert-like. And this gradient is present, throughout the center of the US. Anyone who's driven cross country will be familiar with this. And so it turns out that depending on the amount of water that's available to a plant, it will end up forming different microbial communities in its roots. So this is a cartoon representing the breakdown of a plant microbiome among all these different bacterial phyla. And the outer circle represents the composition of a root microbiome when there's plenty of water, and the interior represents the microbiome composition under a drought scenario. So you can see that something about the plant's response to drought is going to affect which microbes are able to colonize the roots. And so I want to relate that process to the process of plants adapting to drought conditions. So we think about cacti and other very drought adapted plants, and they have some traits in common that allow them to perform well even with minimal water availability. So things like traits that will stop water from evaporating as much out of their leaves, like special forms of photosynthesis, having really thick leaves, and also like reflective leaves to lower the temperature. So those are sort of adaptations that plant species can evolve, but also within an individual plant, there are sort of behavioral changes or phenotypic plasticity that can happen. So when an individual plant starts to experience drought, it has this physiological response where it closes the stomata. It can invest more of its resources into root growth to try to find more water resources. It can increase the kind of waxy layers on the leaves that will prevent water loss and so forth. And an important point here is that many of these traits are traits that can be affected by the microbes living on the plants. So people have done experiments showing that when you inoculate plants with a certain group of microbes, a lot of these traits can change. Okay, and so I'll just throw up some pictures here of the different study sites. So what we did is we went across this precipitation gradient in Kansas, and we collected soils from these 6 prairies, and these were all pristine prairie sites that have never been plowed up for agriculture. And that's important because we wanted to get these soil communities in kind of their natural state, particularly as it involves precipitation. So you can imagine these easternmost soil microbial communities evolved under conditions where they kind of had plenty of water most of the time, whereas these more western soil communities are full of organisms that have had to adapt to really drought desert-like conditions, as you can see here. Okay, and so these prairie soils have never been irrigated for agriculture, which is why we think that they're good representations of that adaptation. And I'm going to talk about some experiments that use these 2 plant species. So here's maize, which is what scientists for some reason call corn. And then on the left here is a plant called eastern gamagrass. And this eastern gamagrass is a US native plant, native to the prairies of Kansas and other places on the continent. And importantly, it's a close relative of maize. It's not the plant from which maize was domesticated, but it is the closest evolutionary relative of maize that is native to North America, or sorry, to temperate North America. And you can kind of see the resemblance a little bit when you look at the female flowers here. Both of them, I think, make some really beautiful flowers. But anyway, so what we did is we did an experimental evolution of these soil microbiomes themselves in the lab. So we took these 6 different soils from the prairies and we made them adapt in the lab for 5 months, and under one of 2 conditions. We either gave them a drought treatment where we withheld water, or we gave them all the water they wanted, okay? And in addition to those 2 treatments, we repeated them, but we added in a plant host. And so now we have these 4 different sort of evolutionary contexts that these soil microbiomes had to deal with. So each of these 6 soils were divided into 4 treatments. And then we just let them go for 5 months. And then what we're interested in is the changes that happened under each of these conditions, because the answer to this is going to tell us a little bit about, how does the presence of a host influence the way that these microbiomes adapt to drought, okay? And so one of the ways we're looking at this, and unfortunately the data are not all back yet, one of the ways we're doing this is we sequenced all the DNA in these original 6 soils before the experimental evolution. So we have this baseline reading of all the different genes present in microbial genes present in these 6 soils. And then after the experimental evolution, all 4 of these treatments, we sequence them again. And so what that will tell us is, oh, sorry about the blurry cartoon there, but what this will tell us is, which microbial genes ended up becoming more abundant in this evolutionary process when the soils were forced to adapt to drought conditions. Okay, so we might see certain microbial genes becoming more abundant and others becoming less abundant. We can do the same thing on this other axis here, this horizontal axis, saying, okay, which microbial genes are increasing in frequency when there's a plant host present, which would suggest that those are genes that improve microbial fitness in the context of being able to colonize a plant, okay? But what we have the most data on so far, and what I think is the coolest part of the story, is thinking about how does this evolution of these microbial communities affect the phenotype of a plant, all right? And so once again, we've got these 4 different evolutionary conditions. So for example, with a host and with plenty of water. And then we have the other 3 combinations as well. And what we actually did to measure, the effects that this evolution has on a plant is we took the outputs of this experimental evolution and we used them to inoculate a brand new generation of plants. So this crude cartoon is showing pots, with a little plant seed in it. And we can take the experimentally evolved microbes and just transfer them into a new pot and grow up a plant in each of those pots. And then we can measure how well the plant performs. And because of the way we designed this experiment, we can actually infer that these any kind of systematic differences in, for example, plant height between the different types of microbial communities is caused by those experimental evolution treatments on the soil communities. And so you'll see this word pop up a lot, the phytometer phase. So if you break this down to phyto and meter, it's just like it sounds. We're using plants to measure the function of a microbial community. All right, so what kinds of data did we get out of these phytometers? Well, we used RNA-seq for one thing. I don't have those data, so I won't talk about it, but that's essentially where we isolate the RNA from plant roots and then sequence it. And what that tells us is not not what genes are present in the plant, but what genes are actually being actively expressed in the plant. And so that will allow us to look at how these plants are responding to the different microbial communities at the molecular level. We're also looking at aspects of nutrient acquisition. So it's possible to measure the abundances of different types of ions. So like carbon, nitrogen, silicon, potassium, phosphorus, all the different components of biological tissue, we can measure them and say a little bit about sort of the nutritional value or the nutrient status of the plant. We can even directly measure photosynthesis. And so we're interested in whether these microbial communities will have impacts on the rate at which the plant can do photosynthesis. We also uprooted all the plants, and we actually used a flatbed scanner to get images of their root systems, and then used kind of this fancy software to, and computer vision techniques to measure all kinds of things about the root system. So for example, the average branching angle. So, thinking about these main roots and then the smaller roots coming off, the computer can actually quickly measure the angle at which those roots are joined. Things like that, the total length of the root, the average width of the roots, etc. And all of that tells us a lot about the kind of the investment and the strategy that the plant is employing to try to find water. And finally, the root anatomy. So when we take a cross section of one of those roots, again, using computer vision, we can actually put numbers on a lot of things about the inside of this root. So for example, we can count the number of metaxylem. That's these circles, these main kind of water moving conduits that are running up and down through the root. The total area of this center part of the plant, we can measure how many cell layers thick are the roots, which will have an effect on water loss, etc. So all of these things got measured for all of our phytometer plants, which gave us a really awesome data set. And so I'm going to present these data in units of, in percentages. So what we did is for each phytometer plant was either given a drought treatment or a well water treatment. And what we did is we asked, okay, for the drought treated plants, what percentage of the control was the plant able to achieve? So let's imagine, like total biomass or something. All of these traits are represented relative to the control. So in this example, the plant would have an identical phenotype to the well-watered control, indicating that this plant had perfect drought tolerance. There was no growth penalty to due to the drought treatment. Okay. In contrast, a plant with a trait value down here would have only 85% of the biomass of the well-watered control, indicating that it took about a 15% hit from the drought. Okay. And so I'll show the data in this format where each of these points represents the plants that were grown with one of those four microbial community treatments. So the microbiomes that were evolved in dry conditions versus wet conditions, and then with a host or without a host. All right, so the purple points will be the plants with no host that got microbiomes which were evolved without a host in the previous generation. Okay. So the moral of the story, after we looked at all of this phenotype data, is that it turns out that the soil microbiomes can improve the drought tolerance of the host plant, but mainly when they were only in the combination of having evolved under wet conditions and with a host present. Okay, so here we're showing we're comparing the plant growth when inoculated with the soil microbiomes that were evolved just under dry conditions. Whether or not there was a host, these those growth rates, or sorry, those root masses are not statistically different from each other. So it didn't matter whether or not those microbiomes evolved with a host if it was really dry while they were evolving. But in contrast, we look at what happens when we inoculate the plants with microbiomes that were evolved under wet conditions. And you can see this really big difference in plant performance between the microbiomes that had a host for the last 5 months versus the ones that were just in a pot with only soil and no plant present. So what this says is that this microbiome adaptation to the presence of a host can happen really quickly when there's ample water. And it can happen really quickly, and it can have really dramatic effects on the performance of the host plant. We saw the same thing. So what we just saw here was root mass. Over here is shoot mass. Okay, so we're seeing the same pattern. And we saw a similar thing with the total root length. So this was using our root system scans and adding up the length, the total length traversed by all of the roots together. We see the same thing where there's a big benefit to having a microbiome that has been evolving with plants and in well-watered conditions, whereas there seems to be no effect of the host on the evolution of the communities when evolving under dry conditions. Okay, same thing with the total maximum root width. And here, looking at the interior of the root, the metaxylem area, so the total area that is covered by these water moving conduits. Here we see, really, we should think about it as soil microbiomes that have been evolving with no host end up having a negative impact on the metaxylem area when they do encounter a plant. Okay, and same thing with the steel area, which is the total area of the central root bundle there. And not all the traits that we looked at showed that same pattern. For example, we saw actually the opposite for water use efficiency, which is related to the photosynthetic output of the plants, where we actually saw that the microbiomes that evolved with the host decreased the water use efficiency of the next generation of plants. Whereas other traits, so here is the cortical cell layers, so that's the number of cells that form that perimeter of that root cross-section. That one just had absolutely no response at all to any of these microbial treatments. And so in parallel, so all the data that I just showed was from that eastern gamagrass species. So this is this wild grass that has been evolving in these prairie ecosystems for a very long time. We wanted to know whether we would see the same thing in a completely different species of plant. So we did in parallel, we did the same experiment, but we grew maize seedlings, okay? And interestingly, all of those really strong effects on the phenotype that we saw in eastern gamagrass did not show up in maize for the most part. So for example, the shoot mass that we saw earlier, the water use efficiency, total root length, all of these were not statistically different from each other, which basically tells us that maize simply did not respond to these microbial communities that had been evolved with or without eastern gamagrass. And so it's really curious, we're very curious why this happens, and we don't have a super clear answer yet, but we think it could have something to do with the fact that these microbes, because they originated from tall grass prairie remnants that have never been plowed, that means that these microbial communities were completely naive to maize. There was no shared history of evolution between that plant species and these microbes, whereas that's not true for the gamagrass. So we think that the genotype of the eastern gamagrass was likely shaped in the presence of this type of microbial community, which must have had some effect on the way they interact with each other. So I'm going to stop there. We're about at the end anyway. So just to sum things up, if I had to boil down all this data into kind of a simple table, this is what it would look like. So evolving these soil microbiomes with a plant, improved drought tolerance of the plant, but only under wet conditions. So why did this happen? Well, we also don't have a very clear idea yet either. Maybe once we have all of our sequencing data, that will help give us some clues about which microbial genes are changing here. It could have something to do with the fact that under wet conditions, there's likely to be a lot more resources. It's less stressful for both the plants and the microbes, which could increase the rate of evolution because reproduction is faster, growth is faster, and as a result, we would expect there to be more evolution, period, in those wet conditions. Okay, so that's one hypothesis that we're trying to explore. And notably, this benefit of having the co-evolved microbial communities does not necessarily extend to other plant species. So these communities evolved with gamma grass, but had no effect on maize phenotype. So we have a lot left to learn. I can answer any questions about our sort of next steps if anyone has them, but I'm going to stop here and just thank my whole lab group who have been really wonderful and done a lot of amazing work on this complex project, and I'm excited to see what they accomplish next. And I'll leave you with this beautiful prairie painting as a nice backdrop for questions.
Awesome. Thank you so much, Maggie. Yeah, this is a great time. There's a Q&A box that you can type your questions in. We already have a couple coming in, so I think Amanda's going to lead us in our Q&A session.
Yeah, so awesome talk, Maggie. Thank you so much. The first question we have, it says, could you say something about the potential separation between existing microbiome plant relationships due to micro and macro environmental climate change?
Okay, so if I'm understanding correctly, this is a question about how is climate change impacting kind of the relationship between plants and their microbiomes. Did I hear that correctly, more or less?
I think so.
Okay. Yeah, that's a great question. And it's totally relevant to what I was just talking about, because you can think about climate change having direct impacts on the microbial communities that are living in the soil, right? Like you think about soils that are in drought prone habitats, maybe they're spending a larger proportion of their time under drought or in the dry stage, and that's going to cause it's going to change the evolutionary pressures on those microbes. It's going to make it even more important for them to evolve adaptations to drought. And same thing is happening for the plants. Now, one interesting kind of aspect of this relationship is that the microbes can evolve a lot more quickly than plants can. Many plants have one generation per year or less, whereas microbes can have many, many hundreds of generations per year. And so there's actually been a few studies that show that the rapid evolution of plant-associated microbes can help plants sort of tolerate the stresses that are coming with climate change a lot more quickly than the plant can evolve on its own. And so there's been several studies that have showed that. There's also been studies, though, that show soil microbial communities being surprisingly resistant to the influence of climate change. For example, I know of a couple of data sets where there was like an experimental warming where researchers took like areas of soil and like artificially heated it, to degrees that we would expect in the coming decades. And another one that where they actually removed snow prematurely every year. This was up in the Rocky Mountains. And both of those showed very little response of the soil bacteria in response to those. So now they may or may not be representative, but it's an active area of research trying to understand how each of those partners will react to climate change.
Awesome. Yeah, so the next question, it says, has anyone looked at microbiome differences between plants with storage roots / tubers and those without? Do you suspect the microbiome differs based on the starch content of the roots?
Oh, great question. I do believe that people have worked on that. However, because I don't work on that, I am not well positioned to tell you much about that other than that there are differences. Yeah, so you're exactly right. That starch content inside the root, that's a major potential food source for these microbes. And so you're going to end up getting microbes that specialize on degrading, being able to eat those long, complex starch molecules, because that's in contrast to the much simpler sugars that are being kind of leached out through the roots. So yes, I know that they have found differences. Unfortunately, I can't tell you in a lot of detail what the differences are specifically.
Awesome. I have a question. Do you have, off the top of your head, like, did y'all notice a lot of differences in how the fungi versus the bacteria responded to drought response in these two plants?
Yes, yes, and what we saw was pretty consistent with what a lot of other people have reported from similar work. So we saw a much stronger shift in the bacterial communities in these soils and roots, a much stronger shift in the bacteria, a much weaker shift in the fungi. And so it seems that the fungi are better able to kind of weather the storm, so to speak, and they are able to sort of keep their community stationary, for lack of a better word, in the face of drought. And that seems to be a fairly common but not universal pattern in a lot of studies that have looked at this.
Awesome. Thanks for sharing that. I have a question I'm going to throw at you, Maggie. I'm not sure that I don't know that you've ever worked or know anyone who works with this. Someone asked if you're aware of any similar microbiome studies of Sarracenia. We work a lot with Sarracenia, the American pitcher plant here. And like, has anyone looked at the microbiome of Sarracenia or their roots?
I mean, usually questions of this nature, the answer is usually yes, because there's just so much out there. I personally, I couldn't say any specific studies, although I feel confident that someone has looked at this. And what an interesting study system to look at. The microbiome inside a carnivorous plant. Yes, the all kinds of factors there that I think would make that a really interesting system. You've got like the high pH, you've got the nitrogen coming from the prey organisms, all the microbes that may be involved in the breakdown process of the prey. Yeah, I'm sorry, I don't have any details other than to say I'm sure that's out there, and I'm sure it's really cool.
Awesome.
Awesome. I have one more question
Go
before
ahead,
we
Maria.
end. This is just out of curiosity, but hypothetically, if you removed all of the microbes from a plant, like on the plant in the soil, kind of starting from like square one and regrowing it, what kind of effects do you think that would have on a plant? And its ability to recover. Like obviously, the plant and the microbes are very reliant on each other in some sense.
Totally. That's a great question. So we've actually done this with young corn plants, and I don't have a picture of it on this PowerPoint, unfortunately, but essentially we removed all the microbes from the seeds and then put them in this like basically like sterilized sand inside a sterile like transparent plastic bag and sealed it and gave it like a sterile water and then just saw what happens. So and people have done this in other species as well, not just maize. So the short answer is that it depends on the plant species. So certain plant species and also certain plant genotypes within species are more reliant on microbes than others. So it turns out that our corn loved it when we grew them without any microbes at all. But I've read about other plants that end up suffering a little bit relative to the controls that have microbes. But yeah, you do see a good amount of variation, both depending on the plant species and also depending on what microbes you're comparing it to, right? Because there's certain, one soil microbiome, as we just saw, could have a one effect on a plant, and a different soil microbiome has a totally different effect. So it all has to be relative to those different communities.
Awesome. That's great. So that's the last question. I'm just going to share my screen really quickly. Let's see. So thanks for coming, everyone. Everyone can see my screen, correct? Yeah. Hi. You can find a recording of this webinar on our website. And our next Science Cafe is going to be next month on the third Thursday of the month on the 20th at 6 p.m. Eastern Time with Dr. Peter Wilf, professor at Penn State in the Department of Geosciences. And he's going to talk about his work with fossilized plants and how they tell the evolution of our planet and reveal landscapes from our past to our present. So that's going to be really fascinating as well. And yeah, if you have questions or suggestions, feel free to email me, my email here, and then feel free to check out our website and our department and follow us on Instagram. Okay, great. And with that, I think we'll give a round of applause to Maggie, and we'll see everyone next month. Thank you very
all
much.
for tuning in, and thanks for the invitation. This was fun.
Yeah, awesome.
All right,
Okay,
have a
bye
great evening.
guys.
Stop the recording.
Yeah.